Thank you very much for the kind introduction. I hope you can all hear me. And I would like to start by thanking the organizing committee for giving me the opportunity to speak twice. And as I'm speaking twice, I also elected to choose the opportunity of being given the chance to give a keynote talk, to talk about something perhaps a little different, raise some questions and open in many ways, but giving you the opportunity perhaps to pick up on a few themes as the conference progresses for the next 3 days. So, as I was just said, I have a bit of a passion in as far as new technologies are concerned. In part, that's a consequence of being involved with new technologies at AstraZeneca for a number of years. But in the context of this, I have become acutely aware of the fact that when it comes to chemistry, most chemists design molecules on pieces of paper. And neither you or I will probably appreciate more than anything that this is a three-dimensional world, and therefore doing chemistry in 2 dimensions is maybe not quite the right thing. Or is it? But let's talk about big data, let's talk about compounds and ways of doing drug discovery in many different ways. So when it comes to it, ever since the new age of automation, big pharma have been able to collect many data sets, and the technology has progressed to a point where numbers are not a problem to be crunched anymore in silico, or even automatically generate them in terms of the technology which has progressed since then. So what actually happens, in my opinion, is that we are now reaching a point of actually reaching saturation of data. And with that comes the question as to whether actually generating more data is only going to increase the haystack rather than giving us more opportunities as a whole. So looking at this a little bit differently, data is great, and looking at greater data is always important. However, what we use on a daily basis is not data, it's information. And when it comes to it, I have a question in my head that I haven't got an answer for, and if you have it, then please let me know. I don't think that actually over the last 20 years we have been able to keep pace with our understanding and turning data into useful information for decision making. So in actual fact, we, I think, generate a lot more data than are actually useful to us. That doesn't necessarily mean that it is wrong to generate the data, but I think the information content within it is something that should become subject to a lot of studies. And with that in mind, I just wanted to give you an example. The example I would like to give you is chemical space, which was in many ways the latest frontier which we all started to join chasing in the mid-90s with combinatorial chemistry. But it also functions to me as an opportunity to ask the question, what actually is chemical space? How can we use it in our favor in terms of drug discovery? So when it comes to it, chemical space can be defined in any number of ways, and I've just given 3 examples that I would like to talk through very briefly. The first one is you can define chemical space through biology, because we're all drug hunters, and at that point in time, biology is always our target and our focus. There are opportunities of using chemical technology and also in silico methods in order to join chemistry to biology in a more direct way, and that's mostly done through the use of computation. And finally, you can define chemistry space just entirely focused on the molecules, and you can define chemical space with the vast number of individual molecules you can come up with in virtual situations through the combinations of the atoms that make sense. However, the important problem with this is you have this vast repertoire of information and this vast repertoire of numbers, and how do you use it constructively? Looking at chemistry space in a little bit more different ways, then what actually has changed in the context of drug discovery since we have started to use automation a lot more, which has actually been given us the opportunity to explore chemical space in many new ways. Automation has had impact not just in terms of synthesis, but also testing, and with that came the invention of high-throughput screening and the opportunity to proceed in many different new ways that are going to give us opportunities to interrogate new targets, new biologies that are actually considered to be the higher hanging fruit, the ones which are eluded us so far because of the difficulty of actually prosecuting them in chemical terms. With this, we have generated large collections that are used in pharma contexts in order to do screening. An alternative project that was originally conceived as a means of actually going away from big numbers to the quite opposite, which actually small numbers of molecules and small molecules with that, is the introduction of fragment-based drug discovery and fragment-based lead generation approaches. They are fundamentally different and fundamentally opposed in some ways to high-throughput screening approaches, because this is a more minimalistic approach that is actually trying to make use of small numbers of molecules to charter out binding sites. When you're looking at it in other ways, in my opinion, combinatorial chemistry and high-throughput screening are more like evolutionary processes that have actually been making a lot of progress over the last 20 years. And with the introduction of DNA-encoded libraries and macrocyclic libraries, the latest introduction to this field, we are now able to generate a number of molecules which is almost begging us believe. And the question is, what is actually quality in this context? Because at the end of the day, the whole use of large numbers of molecules introduced a very useful and very insightful debate into the question, what is drug-like and what is quality in chemical terms or in drug discovery in general? And I think that is a debate which I think is hopefully going to keep us going for a while yet. In the context of this particular discussion, I would like to compare fragment-based drug discovery more with a revolutionary rather than an evolutionary process. And the particular reason why I thought that this is an opportunity for differentiation is that combinatorial chemistry and high-throughput screening have used opportunities to grow, whereas fragment-based drug discovery asked the question and then technology had to come up with an answer to make it happen. So in some ways, it was not necessarily something which would grow slowly through various iterations, but it tried to ask a question in new ways. And with this, again, came an introduction of a new term, which is efficiency in chemistry, which, again, I think is very important because it helps us to maintain a focus. So overall, we have revolution and evolution. The question is, are there opportunities for the future? And if you look at chemical space again, most of the time, chemists look at it in a context of structure-activity relationship and properties. And that is very useful and very helpful and has led to interesting new drugs, as you've just heard from Professor Lam. Alternatively, though, I think there are opportunities still missed out, and one of them is to consider what actually is at the heart of chemistry, that is the molecule, and describing the molecule, which is obviously very difficult in many ways, because this is something that has been done for the last 100 years or so, ever since the invention of quantum chemistry and the quantum mechanics that came along with that. So conformations, is that a new dimension that could be explored? Is it an opportunity? And I would like to take a leaf out of one of the businessmen that I really respect for what they have to say in terms of opportunity. And in that sense, I would like to consider and liken the opportunity of conformations as a new way of describing chemical space, more as a revolutionary process rather than an evolutionary process, even though a lot is already there. Because in my opinion, there aren't yet good enough and sufficient experimental methods available to define fully what molecules do in terms of their behavior in solution or in the context even of a bound target. So when it comes to it and conformations, so far I think the key points that have been pursued are numbers, and numbers are very crude, and in some ways they are almost like a big hammer to crack a small nut. But when it comes to the use of molecules and understanding molecules that we are working with on a day-by-day basis, then I think there is a lot of opportunity for seeing the beauty within, if you start looking and scratching under the surface of what molecules actually do. And in this context, I would like to go back and take a leaf out of somebody's book, which is now nearly 20 years old. Reinhard Hoffmann coined a phrase in some of his work, which was around conformational design. And I think it is important to consider at least whether or not the study and understanding of selection processes that are actually going to make it possible to make molecules with a single shape As we've seen in the preceding talk, molecules bind in a specific way to biological targets. However, when it comes to it, conformational space, which is the space that chemicals target or uniquely explore in solution, is not a single entity. So what other things does conformational space offer us then when it comes to the concept of chemical space? Well, when you look at histidine, for example, it is a collection of atoms which have a particular connectivity, and with it come potential opportunities in terms of charge states. Broadly speaking, it is arrangements of different types of pharmacophoric points in a molecule. When you now overlay on top of that the opportunity of actual flexible motion within that molecular framework, you quickly arrive at a situation which is described by data from the CSD in the following way. The molecules, particularly the Chi-1 and Chi-2 bonds, and I've taken a nomenclature from peptide chemistry, are actually observing multiple states in the solid state. Now, if you follow me and accept my hypothesis that the small molecule solid state properties are actually reflective of minimum energy states and therefore reflective of behavior in the solution, then you can clearly see that histidine, with its motion, goes through various different states, various different conformational states in solution as well. And sampling these, it arranges the pharmacophoric points in very different relationship relative to one another. So in this case, you have 3 times 2 conformations, which in my opinion accounts for about 6 different pharmacophores that the one molecule, which is reasonably small, is actually describing. Now multiply that by the number of bonds that you have in larger molecules, drugs, which we have seen some examples of today, then you quickly see that conformational space is actually a lot larger than perhaps one would think of initially just looking at the molecule. And therefore, in my opinion, it actually does offer opportunities of looking at chemistry in different ways. So when you look at histidine, I just try to enumerate all of the options for the conformational sampling that this one molecule takes in solution. Now the other question then is, if molecules do move through different conformers, how many conformers are there? So I had a look in the literature to see what kind of flexible molecules I can find which have been studied in terms of their conformational space, and I came up with 1-octene. 1-octene is characterized by 5 bonds which connect sp3 hybridized atoms and 1 which is connecting sp2 and an sp3 atom. So if you're looking at this, I wonder how many of you would recognize this as potentially quite a flexible molecule. Now, I will spare you the question of raising of hands here. But when it comes to it, molecular mechanic calculations and ab initio calculations have been performed to characterize the motion of this particular molecule. And admittedly, this is work of the 1990s, so it is quite dated. But then it raises the point of people being interested in this a long, long time ago. And when it comes to it, the molecule can describe 131 conformation as predicted by in silico methods. When you're looking at the experimental evidence for conformers in the solution state, you see that there's only experimental data to support 15 conformers for 1-octene. Now, I don't know whether the experimental methods are not good enough to detect the missing over 100 conformations, or whether in silico methods get it slightly wrong, and then there is something more to conformational behavior of molecule that perhaps meets the eye initially. So the way I like to look at molecules these days is not necessarily as a fixed connectivity of atoms, and this is something I again borrowed from the internet, so excuse the fact that that hydrogen here is yellow rather than white, but that's the way it was depicted on the internet. The key point I was hoping to show with this is something that is quite important to me. Conformers, and the way I look at conformers these days, is not actually a collection of closely related energetic states. In my opinion, there are only a small number of conformers which are really relevant, and then there is a lot of small motion around these particular conformatic states. So when you look at this molecule, you can see, hopefully, what I'm trying to explain, that a molecule moves backwards and forwards between particular conformer states, which can be potentially described by one mean angle. And then it flips to another conformer state, and again it wobbles about. So there is entropy in molecules, and that isn't something which is surprising to us, because we know that entropy is a key component to thermodynamics, and that's what our playground is in chemistry. So when you take that idea of molecules going through mean bonds with a particular spread of angles around it, then you all of a sudden see yourself in the position of describing bonds in different ways. Here I would like to just point out that I've taken something again from the internet, which is a collection of curves which are Boltzmann distributions. And what I would like to do is using the Boltzmann distribution as a means of explaining actually the link between probability and energetics. And that's what Boltzmann's equations do. Using clever mathematics and opportunities of actually looking at partition functions that are always inherently important to be defined, then you can look at connecting energy and probability together. So in the context of a bond, then it becomes evident that you can actually describe a bond as a mean torsion, which is representing a particular conformal state, and around it you have a torsional flex. And depending on the degree of control you have within the actual framework that defines the particular torsion, the mean torsion you're referring to, then you have more or less conformational control, which means that you have either a sharp or a very broad probability curve, and with that come a lot of angle flex or very sharp angle flexing. The importance of that is potentially quite clear when you come to molecules in the bound state. If a flexible molecule binds in a rigid pocket, that's not good. It's like trying to put a round peg into a square hole. Alternatively, if your protein, which is always flexible like most molecules are, has particular flexibility around a particular binding site, and your molecule is very rigid, it's not going to fit either. So you have to have a match between the dynamics of the protein and the dynamics of motion of the small molecule in order for tight binding to actually occur. So if you want to see the kind of picomolar affinities that you've seen described earlier for factor 10A, then I think you have to have not just a match in terms of shape, but you also have to have a match in terms of dynamic behavior of both small molecule and the target around it. Or don't you? So, coming to the question of what chemists make of chemistry. And these are examples I've been taking from my memory of how discussions have taken place over the years in the context of designing new molecules for projects. So sometimes chemists drew up a molecule in this sort of way and basically started designing around it by moving atoms and bonds in keeping with the understanding of SAR and property space, of course. So this is not just willy-nilly putting atoms in certain places just for the sake of it. There was a hypothesis linked to that, of course. However, if you look at a molecule like this, it's not three-dimensional, and the world is three-dimensional. So somewhat more sophisticated chemists then started to draw molecules a little bit like this, and yet they still have forgotten key points, because you can look at ionization states, you can look at the individual parts of the molecule, and then you still have information that you can include in your even two-dimensional depiction to represent the chemistry that you're interested in. So the really clever ones, they drew molecules like this, in my opinion. They put charge states right, and they also try to define, well, actually, there are molecular parts which come towards you and which go away from you, and there are conformational preferences, i.e., you want the proton here to be in line with the proton there, don't you? So that's, in my opinion, the playground where chemistry should take place, but quite often it's where we started from, that chemistry starts and in sometimes, unfortunately, finishes. So what that actually means, in my opinion, is that when it comes to drug design, depending on where you are on this continuum, and I'm sure you can progress this further to the right if you wanted to, starting to go, for example, in silico and look at molecules in 3D, then your information content that you use for drug design becomes more and more sophisticated and more detailed. And with that, potentially comes the opportunity for making better quality decisions. So I'm talking here about decision making, not necessarily better drugs. But in terms of decision making, The better decisions are the ones which are the informed decisions, not the ones which you kind of just go about and go and decide quickly. So seeing is believing, is it? Depends on your perspective in chemistry. Well, do you see the spider or don't you? It depends on your perspective. Now, to wrap up, some quick examples of where chemists have gone wrong, in my opinion, just looking at chemistry in 2D. The bioisosteric question, amide versus sulfonamide, they are the same, of course, aren't they? Well, if you look into the literature, they are, because you can find literature which actually says they're bioisosteric of one another, and if you don't believe me, that's there and there, they're in the same table, they are the same, of course, biologically. But it very much depends on your perspective, as always in life. And the way I try to look at molecules, as I said before, is more in 3D. And then all of a sudden, when you look at data from the PDB and the CSD, you see that sulfonamides behave differently to amides. They populate, when it comes to this torsion between the sulfur to the phenyl or the carbonyl to the phenyl, they populate different areas of chemical space. So in 3 dimensions, they are different. So when it comes to it, don't be a lemming, don't follow doctrine, don't follow dogma, but actually try to make a decisions based on information from multiple sources. So you call on data to get information to make decisions. Otherwise, you end up in a situation where the focus in drug discovery, which is very high these days because of the targets we all set, provides you with blinkers because you have focus and you have dogma. And the two things together, they are, in my opinion, are a lethal combination. And why is that? Because the more blinkered your views are, the less you see. And if you are just by chance lucky enough and your perspective changes at the right time, well, actually, you get the surprises that you need, don't you? So, however, the data that you need to consider is also important in terms of the usefulness, the appropriateness, and therefore, again, you check your hypotheses. That's the mantra in some ways that has come out from this. So looking at the PDB, here's one example of a molecule which is deposited in the PDB, and it doesn't look pretty because sp2 atoms have a 3-dimensional shape and ring pockets are in the wrong state. And is that a single example? No, unfortunately not. It's about 50% of the total PDB content where you have a problem in terms of the actual way that the molecule is fitted to the data, to the density of the molecule. So again, the question is, what is the problem here? Well, it's Chinese whispers. You take the information as it is, the histograms as I used them before, and just make decisions on them. And in the end, you end up with a situation of Chinese whispers. You're making inferences about something that you really can't make. And in the process of this, you start to develop blinkered views. And unfortunately, it doesn't quite work for you sitting as far back as you are, because if you start looking at this and focus on this cross, that blue dot disappears because you have a blind spot on your retina where the nerve is hitting. And therefore, be wary of what data you use. Test the hypothesis first whether it's useful. But conformational design, can it be used? Well, let's look at nature. Nature I like because nature is always right and nature is a great teacher. So in nature, you find polypropionate drugs, and I've worked on them as part of my PhD, and they're really hard to make. But nature has no problem with it, and nature uses a lot of controlling factors to get shapes and conformers right. And that's something we should really be looking at. And in this one molecule, you can find the whole litany of all the things that you can use to find conformations. And is it actually helpful? Well, if you look at the maths, it is, because you can save about 1.5 kcal per mole if you fix one rotatable bond and get it into a single conformer state. I'm not talking about a ring here, I'm talking about a single conformer state. And does it work in action? Given the time, I'm going through this more quickly now. So it does. This is an example from orexin 1, and I put the build on. So you have molecules which were modeled, and then NMR data was used in order to refine the decision-making on which is the most probable conformer. And that was subsequently confirmed with small molecule X-ray, and then the chemists went in the lab and made the ring molecule to prove it, and it all fitted together. That's what I'm talking about in terms of 3D design. That's what I'm talking about in terms of trying to make sure that you use all the information built within the molecule that you're dealing with. Look at them, probe at them, and get the different perspectives on this. Here's another example. One which I like very much because it is one about the question of the central bond. The central bonds here are very flexible, and therefore what chemists often do is they make ring structures to constrain them, which is fine, and it serves the purpose. 1 micromolar, 16 nanomolar, Bob's your uncle. Off you go, good day in the lab. But there are other ways which are a little bit more difficult, perhaps, but they are also giving us opportunities. So this here is a molecule which is fully optimized, but it's different. It's a different way of optimizing. It's not rigidity through ring formation, it's rigidity through conformational selection, using what nature has used in terms of atom type changes, hybridization changes, etc., in order to make a molecule not more rigid, but select out the right conformer. And it works as well. And that's a week in the lab. So the concluding remarks really from me are simplicity rules. There's a lot of information out there. There's even more data, but keep it simple, please. Because the question is, can you describe something in simple words, then you understand it. And that's what we're all about. We're all about understanding. We're all scientists. So find X. What do you want to use? Well, I take the simple approach. It's there. So Occam's razor, in my opinion, is one which is really simple and really important. If you have two solutions, one complex, one simple, go with the simple one first, and then make it more complicated if you have to do it. So all the technology we have today, it's useful, but perhaps not quite the point where we should start. Go back to principles and use those for drug design. And as I'm running out of time, I want to stop here.
Thank you for the talk. We have time for one question only, and then we can talk over lunch, maybe.
Keep it simple, go with the closest.
Thanks for a nice talk. You were talking about conformations and flexibilities of molecules, but basically the conformations and the flexibility or the plasticity of the targets is one of the issues which is probably equally as important.
Yeah.
And how do you include that one in your predictions?
I would like to say that this is why I'm keen on looking at conformational selection rather than rigidification. So if you play with molecules in different ways, yes, you can constrain them by ring formation, which gives you very rigid entities. But if you try to look at conformational selection through sterics and electronics, which are not as rigorously controlled and as tightly controlled, so going back to nature, then that perhaps gives you the flexibility in the bond to go along with the flexibility in the target.
I understand that, but the flexibility of the protein target is probably much more important than the flexibility of the small ligands.
Yes, it is, and how you skin that cat, I have no idea, because dealing with small molecules is hard enough, never mind connecting them all up to a macromolecule of the size of a protein. So perhaps it's got to go back to simulations in the computer to understand that and fit that into the data that you have from crystal structures, B factors, etc. So that's another overlaid problem, which I think is perhaps one bridge too far at this point in time.
Okay, thank you. So thank you again.